Hi everyone, you're welcome to this YouTube channel where we throw more light on biotechnology. As we know, it is the applications of the genetic material from living organisms to make a desirable product. But how do we actually introduce this genetic material into another organism is what we're going to explain today, which is known as the ligation of gene. To ligate means to sew or to attach the host gene to the genome of another organisms. But it is actually catalyzed by an enzyme called a ligase, which helps in binding the host gene to the gene of the other organisms. It is carried out by the three principles in ligation of gene, which include the conjugation, transduction, and transformation. We'll be explaining this one after the other. The conjugation is such a principle in which the gene of the host is coming as a physical contact with the genome of the other organisms without a mediator. We're not using vector in introducing the gene from the host to that of the other organisms. Why in transduction is such a principle in which we use a vector like virus to introduce the gene from the host to that of the genome of other organisms. Vector in this concept actually means the agent which helps to transfer or introduce the genetic material from the host to that of the other organisms. So there are some virus like bacteriophage which actually infects other bacterium, thereby producing another bacterium. In a transduction, we can see this in a DNA recombinant, in which an organism that's actually producing insulin, should the organism could not be able to produce enough insulin to regulate the glucose level of the body, there's need for sourcing for gene that's producing insulin in such an organism, thereby introduce it into another organisms like bacteria that are not producing insulin. Bacteria like Escherichia coli, and after cultured for a while, you start to produce the needed insulin. So this is carried out as one of the principles in ligation of gene. The last aspect, which is the transformation, is such a principle in which the genetic material from the host is coming as a naked genetic material to the genome of the other organisms. There's no mediator. We are not using one vector in transferring such a genetic material to the genome of the other organism. No, the two, they are coming together as a naked genetic material. But some instances, cell could not be able to pick the desired gene indiscriminately. In such an instance, the cell needs to be rendered competent to be able to pick the gene of interest. And to render a cell competent, it needs to be treated with either calcium chloride or magnesium chloride. After that, next thing is replication, decloning, and so many more. Thanks for watching, and don't forget to subscribe to this channel as we're going to throw more light on other approaches and techniques in biotechnology.